PSA, which include getting access to a wide variety of experts in the field from across Canada, awards from peers and for graduate students, availability to present your work at functions such as the Green Bagger Series or our annual meeting, and access to the Canadian Journal of Plant Science for a discounted rate as well. So if you're interested in joining or know of anyone interested in joining, I would definitely encourage them to register. We are currently looking for presenters for a 2024 Green Bagger Series. So if you know of any students who would like to work on their presentation skills and present their preliminary results or even their proposals, I would encourage them to reach out to Marcy and she can connect with setting up for our 2024 season. With that being said, I would like to welcome our presenter today. Today we have Simranjeet Kaur. She is an international student from the University of Alberta, and she has been collaborating with Agriculture and Agri-food Canada as a research participant. She obtained her bachelor's degree from the Punjab Agricultural University and received the University Merit Scholarship for 3 consecutive years. Her current research project represents a significant stride in the pursuit of knowledge, enabling her to contribute to the world and establish a career in plant breeding and genetics. Simranjeet is dedicated to maintaining her credibility through diligent work. Simranjeet's research aims to combat stripe rust, a destructive wheat disease caused by Puccinia striiformis. The emergence of the virulent strain in 2000 rendered many resistant wheat varieties susceptible, highlighting the urgency to find new resistance genes. Using the advanced mapping techniques and diverse wheat lines, Simranjeet is pinpointing the genes and quantifying their impact by developing molecular markers from her findings. Simranjeet hopes to enhance widespread wheat cultivars, making them resilient against this damaging disease. And with that, I would like to pass it over to our presenter today. So welcome, Simranjeet.
Thank you so much, Alexa, for this introduction. So now I'll start my presentation. So the topic for today's discussion is about nested association mapping to identify stripe rust-resistant QTLs and their markers in spring wheat. So first of all, what is stripe rust? Stripe rust is caused by Puccinia striiformis, former species tritici. It is a heterogeneous macrocyclic fungus. And what I mean by heterogeneous, it means that it needs 2 types of hosts to complete its life cycle, and microcyclic means that it produces 5 different types of spores. Here in this picture, you can see the uridospores, which are yellowish orange colored. These uridospores are present in the form of parallel streaks, and due to which it's known as stripe rust. The other name of stripe rust is yellow rust. It's a polycyclic disease, which means it can complete more than 1 life cycle in a year, which is really devastating. The optimum temperature conditions for stripe rust is 7 to 12 degrees Celsius of temperature, and pathogen loves to thrive in a cool and wet climatic conditions. This slide is about Puccinia pathway. So the pathogen overwinters in the Pacific Northwest United States. So basically, Puccinia pathway extends from Mexico. It travels throughout the United States, and then it reaches the prairie provinces, which are Alberta, Saskatchewan, and Manitoba. And this bumpy area with the shaded region, this is the rust area where most of the rust attacks are being found in the wheat fields. It's been recently reported that pathogen overwinters below the snow cover even on the wheat crop. And as a result, the infection is transmitted to the next season crop. Other than this, various epidemics in Western Canada are also reported in 2005 and 2011, and stripe rust seems out to be the most devastating disease of Western Canada. So there are the 3 objectives. First of all, it's the meta-analysis. The second one is the nested association mapping. And the third and the last one is about development of molecular markers. So here is the first part, that is meta-analysis, where stiprus-resistant QTLs, meta-QTLs, marker trait associations known as MTAs, and genes are represented in this Sarko's diagram. So here the outermost circle, it represents a scale on which various wheat chromosomes are there. Like this is 1A chromosome, this one is 1B and 1T chromosome. Inside it, the outermost circle represents MTAs, that are marker trait associations. And here, the innermost circle is about meta-QTLs. In between them, there are different QTLs that are located based on their physical locations. So about the genes, in total, there are 86 stiprus-resistant genes that have been identified. And other than this, there are some genes that are temporarily named, like YRNAM on 1B chromosome, YRZM 175 on 2A chromosome, and YRAS288R on 4D chromosome. So in total, the 86 stiprus-resistant genes are identified, but on 1A chromosome, no gene has been identified so far. The point to consider here that there are various QTLs and meta-QTLs that are identified on this chromosome, but still no gene has been identified on 1A chromosome. On 1D chromosome, there is just one gene, that is YR25. Some genes such as YR27 and YR7 on 2B chromosome are being defeated by the recent virulent races of the pathogen. In addition to this, on 2A chromosome, YR70 and YR32 genes are defeated. There is a gene that's YR64 that exhibit partial resistance, and it is present on 1B chromosome. The next slide is about the chromosomal location and what I conclude from this meta-analysis. So for the chromosomal location, the highest number of QTLs were reported on 2B chromosome, followed by 3B chromosome of the wheat. And the lowest number of QTLs are reported on 6T chromosome, followed by 5T chromosome. In this diagram, this is basically an upset plot. So here there is CIM, which is composite interval mapping, inclusive composite interval mapping, GWAS interval mapping, multiple QTL mapping, and multiple interval mapping. So these are the various mapping methods that are used that I had seen while analyzing them. So the mostly used methods are CIM and ICM, followed by GWAS. So these are the two methods. Basically, CIM and ICM, they comes under QTL mapping, and GWAS, they comes under the association mapping that we'll discuss in the coming slides. So these two mapping approaches, QTL mapping and association mapping, becomes under traditional mapping approaches. For QTL mapping, two contrasting parents are selected, and from them biparental population is generated, which is subjected to phenotyping and genotyping trials. For genotyping, a suitable SNP system is selected through which linkage maps are constructed, combining with the readings that we get from the phenotyping data by the multi-location trials, then the QTL mapping is done. On the other hand, for association mapping, diverse germplasm is selected, followed by phenotyping and genotyping. But in this case, for genotyping, LD measurement and population structure and relatedness is considered. So LD measurements is like the non-random association of the alleles at different loci in a given population. And for the population structure, it's generally referred to as the presence of the subpopulations within a large set of population. Because for the association mapping, we are using diverse germplasm selection. And there is a need to see that how they are related among themselves. For QTL mapping, there is for this QTL mapping, we are using biparental population. So recombination events are quite limited. As a result, the mapping resolution is not that high, but the detection power is quite good for QTL mapping. On the other hand, for the association mapping, The mapping power is good, but the detection power is still compromised. Or these two methods, they have their own merits and demerits. The next slide is about the nested association mapping, which I plan and I'm still using that in my research. So why we opted for nested association mapping? Because in nested association mapping, there is a common parent which is crossed with diverse resistant parents. And from them, they are subjected to various generations. Generally, we go till F7 generations to generate one NAM population to fix homozygosity. By going for various crosses, the allelic diversity and the number of recombination events are increased, which leads to a balanced QTL detection and mapping power. In my research, we are planning to go for 3 NAM population. So in this, there is 3 susceptible parents too, like Everset, Vesper, and Cardale, which are crossed with the diverse resistant parents to generate 3 NAM population. These 3 NAM population have total 18 real families, and in total we have around more than 2,900 lines that are going to be subjected for this nested association mapping. So the next step after this is the results that we did this year. So the first of all, it's the collection of genotypic data. It involves the collection of leaf samples, the isolation and quantification of DNA, and then their genotyping using SNP. The next step is the collection of phenotypic data. For collecting phenotypic data, flag leaves were assessed 21 to 28 days after inoculation. And after that, the severity and infection responses were recorded. For severity, there is a range from 0 to 100%. 0 means no infection, and 100% means the whole leaf area is covered with stipe rust response. To understand the severity percentage, it's just how much is the infection. For the infection types, it's about which kind of infection is there. So it ranges from 0 to 9, and it's based on the size of pustules, the amount of chlorosis and necrosis. This is the summary of stipe rust data that we collected from NAM population at Lethbridge during 2022. So here in this column, we have the NAM populations, which are 3 NAM population, and the names of the real families, which are in total 18 real families. They have to go for 2 replication. For the parents, there are susceptible parents and the resistant parents. As I mentioned earlier, we have 1 susceptible parents and various resistant lines. So here, DS means disease severity, IR is the infection response, and then the readings from the disease severity and infection response were multiplied together to have the coefficient of infection. This goes for the parents. Then for the populations, it's about minimum and maximum readings. For the minimum and the maximum infection responses were recorded. Through them, we got the mean. So this is the summary from 2022. Unfortunately, this year we didn't get the good stripe rust data to analyze because of the drought, but the next year we surely think that we have something to analyze. Next is the future perspective. So it deals with the analysis of genotypic data. So this is what we plan to do in the future after genotyping. Then construct the linkage maps of each real population and the consensus linkage maps for each NAM population, and then there is a comparison. After that, there'll be identification. Hopefully, we'll be able to identify some QTLs and markers for stipe rust resistance and develop some molecular markers so that we can identify some of the locations which are rich and which can be used to develop the cultivars with stipe rust resistant genes. So here's the acknowledgement. I would like to acknowledge my lab mates and my funding agencies for making this project possible. Thank you so much. And if you have any questions, you can please, you are open to that.
Thank you, Simranjeet. That was a very good presentation. I
Thank you so
think
much.
you have very great presentation skills. It was very easy to follow. I would like to open the floor up to questions now. So if anyone has any questions, please raise your hand or turn on your screen. Yep, Raymond. I guess I had, so what locations are your trials in?
Yeah, my trials are at 3 locations. They are at Lethbridge, Weston, and Edmonton.
Okay.
Yeah.
Any questions from our audience? No. All right. I guess, how did you choose your resistant lines or your susceptible lines? Sorry.
Yeah, because these lines are widely grown in the Canadian fields. So basically, whenever we are choosing any susceptible line, so we want its background, like we want the other traits like the yield and everything, and we want to like transfer the resistance kind of a thing to it. So that's why we chose the one that are being widely grown in the Canadian fields. And so based on that, we chose 3 of them. So that's the reason. Thank you.
I thought it was interesting too how there weren't any QTLs on was it B1 or A1?
It was 1A chromosome.
1A, yeah.
The
Do you
QTLs
expect?
are there. Yeah, the
Okay,
QTLs are there, but the gene is not identified till now.
yeah. Do
Yeah.
you hope to identify a gene through
Yeah,
this
we
work?
hope that, like, to conclude that, I think we need to analyze the genotypic data that I just collected this time. So I hope I will find something there, but I'm not sure yet.
Definitely. No, that could be
Yeah,
very exciting.
yeah, it will be.
One more. Anyone in the audience have any questions for Simranjeet? No? All right. Well, thank you, Simranjeet. That was a really great presentation. As I
Thank
said,
you so
you're very
much.
well spoken and very good at breaking down your topics and explaining it. So I am excited to see where this project takes you, and hopefully you will give us an update when you get some more results in. But thank you to everyone for attending. This was our last Green Bagger series of 2023. We hope to begin them again in 2024. So keep an eye on our LinkedIn pages and on your emails for our 2024 Green Bagger series. And again, if you know anyone interested in presenting, please let them know so they can contact Marcy. But again, thank you for attending and wish everyone a happy holidays, and I hope to see you in the new year. Thank you.
Thank you. Thank you for having me here. Thank you.